Good morning, everybody, and welcome back. So I'm in the lab today. I'm going to be presenting on Lab 10, digesting molecules, and then we're going to set up the experiments and run through everything. So with this lab, we're going to be reviewing some of the macromolecules that were covered earlier in the semester. We'll look at carbohydrates, we'll look at lipids, we're going to look at proteins as well, and then digestion of those molecules within our bodies. So we're going to start off with just looking at the different macromolecules and just kind of what are the characteristics, where are they digested in our body, and what do they form in terms of the monomers and the polymers of these molecules. So starting off with carbohydrates, there are different categories of carbohydrates within our diet. We can have simple carbohydrates. And simple carbohydrates are essentially monomers, like glucose or fructose, so they could be a monosaccharide, or alternatively, they could be a disaccharide. Maltose or sucrose would be two common examples of disaccharides. And we find these very commonly in our foods with respect to the disaccharide sucrose being very, very abundant. You may have heard of high fructose corn syrup. Yeah, that's another disaccharide as well. It's a common sweetener used in our foods. And then we have complex carbohydrates. Dietarily, complex carbohydrates are mostly starch. That's the main component that we would get in our diet. And it's simply the same glucose monomer strung together over and over and over again, hundreds or thousands of times. We can convert these carbohydrates into glycogen in our own bodies. So we'll convert the starch or the monosaccharides or disaccharides into glycogen, and we tend to store that glycogen in our liver, our muscles, and some of it in our brain as well. Looking at a second macromolecule, we have, it's not working. So another category of macromolecules would be proteins. And for most of us, if we think of dietary protein, we think of meat. And yeah, commonly we associate with that. Could be chicken, steak, fish, the white part of an egg, so the albumin of an egg is primarily protein as well. But we can also get protein from vegetables. Dark leafy greens, for example, would have proteins in it. And you may have heard the term complete protein. And so the term complete protein is not really a biological term so much as it is a nutritional term. And what it's referring to is the amino acids which are present within that protein. So there's 20 naturally occurring amino acids, and those are the monomers, or the building blocks of proteins. So the monomer is an amino acid, the polymer is the protein, and we produce, I believe it's 12, could be wrong, I believe there's 12 amino acids that our body can synthesize, and so we have to get the remaining amino acids in order to make our own body protein from our diet. And the term complete protein simply means that it has the amino acids that we do not produce ourselves. And so if you are a carnivore or an omnivore where you eat meat, you'll get those in your diet naturally. If you're a vegetarian or vegan, you might need to diversify and make sure you're not eating, or you're eating a diverse range of legumes in particular, so beans and grains, dark leafy greens, in order to get a complete protein in every meal, or at least most meals, so that you can synthesize your own amino acids. We're going to be using egg whites today as our protein, and so we'll look at that in a little bit when we get things set up. And then our third group of macromolecules that we're going to be looking at are fats. And fats are interesting within our diet. They play essential roles in our bodies. So some of the important roles that fats might play in our body are insulation. So we tend to put fat in areas where we have large blood vessels close to the surface. If you've ever wondered why people get a double chin and we accumulate fat along our neck, it's because the carotid artery is going up, and we want to insulate that so the blood doesn't cool off significantly as it travels up close to underneath our skin as it goes to our brain. We also tend to put fat over here because we've got major blood vessels going out to our arms, and then our femoral arteries and femoral vein going down into our legs. These are areas where we tend to want to wrap them, if you will, in a layer of fat to provide insulation, so our blood doesn't cool off as it goes out to the extremities. Fat also provides cushioning for us. Think about your butts. Every time you sit down, we have, sure, muscle there, but also some cushioning, some padding provided by the fat. In addition, we'll also provide that cushioning along our belly region. Our rib cage only extends from our clavicle down to this region right here, where our diaphragm, or the bottom of our rib cage, and so our lower organs are not protected by ribs. The main reason for that, if you think about it, would be pregnancy. Imagine if your rib cage actually extended all the way down to your pelvis, and you were a female, and you got pregnant, then snap, crackle, pop as your baby starts growing in you. So our rib cage has to stop up here, but we also need to provide some protection for our lower organs. So we do that with a combination of abdominal muscles and then a little bit of fat, providing extra cushioning and layering there. And then we also use fat for energy storage. Gram for gram, ounce for ounce, pound for pound, however you want to look at it. Nothing can store energy, potential energy, as efficiently as fat. So it's a nice long-term energy storage for our bodies. And then finally, our brains. A lot of fat up in our brain, and that's essentially creating what's called myelin sheath, which helps us think better. It creates essentially like a conduit over our nerve endings, and that helps us speed up neurological processes. And so those are some of the functions of fat. In terms of where we get fat in our diet, or what are the different types of fat, well, you've probably heard of trans fats and saturated fats and omega-3 fatty acids. Some are good, some are bad. So we want to try to limit the amount of saturated fats in our diet. Those are the types of fats which tend to cause cardiovascular problems. They can accumulate in your bloodstream, form plaques, lead to arteriosclerosis, which is hardening of the blood vessels, can lead to hypertension, high blood pressure. So we want to try to avoid, or at least reduce the amount of saturated fats in our diet. Other fats are healthier. The omega-3 fatty acids that we would find in fish oils like salmon or olive oil, these tend to be healthy. And then on the left-hand side of the screen, we just have some sources of fat in your diet. So imagine today, you're home, you're alone, you've gone through all of your good food, and you're stuck with bread, bologna, mayonnaise, and Swiss cheese. Yeah, social isolation sucks. But we're sitting home alone, so you're like, I'm going to make a bologna sandwich. So you grab your 2 slices of bread, you throw a couple slices, just 2 slices of bologna in there. We've got about 16 grams of fat in that bologna, 2 slices of cheese on there, another 12 grams of fat, a little bit of mayonnaise for good measure. Put that together, and you end up with a bologna and cheese sandwich with 36 grams of fat. That's the equivalent of 9 teaspoons of oil or fat or lard. You may as well just open up that tub of Crisco, peeled back the lid, got your tablespoon or your teaspoon, and started munching away on Crisco. Good times. Anyway, fat is important for us, but yeah, we got to watch out in terms of how much we get. The theme of today's lab is digestion. And with digestion, the main reason we digest our food is to make the food small enough so we can actually absorb it into our bodies. So digestion is breaking up the food into smaller and smaller components, and then the absorption will occur along our intestine, and then we can absorb that food into our body and utilize the potential energy in that food to do work, to undergo cellular respiration, to synthesize our own proteins, if it's amino acids, stuff like that. So where does digestion begin in your body? Well, it turns out if you had that bologna and cheese sandwich in your hand and you're like, yeah, I'm still going to eat it, you take a bite of that. As soon as your teeth make contact with the bread, you've got digestion. So the first type of digestion which occurs, it occurs in your mouth, and it's mechanical digestion. The mechanical digestion occurs when you're chewing your food. When you take a bite of your food, and you chew it, and you grind it, and you're physically breaking that food into smaller and smaller components. So digestion begins in your mouth, mechanical digestion, but you also have an enzyme in your mouth. We have saliva. And so if you think about your saliva, it serves a couple of important purposes or functions for you. One of those functions is lubrication. So imagine taking a bite of that sandwich, or better yet, a bite of just a couple crackers, and if you didn't have any saliva in your mouth and you put some crackers in your mouth and started chewing them, it'd be pretty uncomfortable. So you're not going to be able to swallow very well without your saliva. Another thing that's in your saliva are buffers. And the buffers will essentially moderate the pH so that if you drink or consume food which is slightly acidic, maybe you have a glass of lemonade, so you don't get acid reflux, or not acid reflux, but you don't get essentially that burning sensation as you're swallowing that lemonade. Lemon juice has a pH of about 2. The stomach acid that you have has a pH of about 2 as well. So imagine drinking lemonade and feeling a burning sensation as you swallow it. Well, the buffers in your saliva help prevent that. And finally, there's an enzyme in your saliva, and the enzyme is called amylase, and the amylase digests carbohydrates. We'll touch more on that a little bit later, but realize that we've got that. Now, in terms of chewing your food, if we ask, well, why do we chew our food? The most common reply is, so we don't choke on it when we swallow. But when was the last time you ate oatmeal? Huh? Did you chew your oatmeal? Of course you did. So your oatmeal is already very small in small particles, but still we have the tendency to chew it. It's our natural instinct to do that. But in addition to making our food particles small enough so that we don't choke on them when we swallow, another important thing that chewing does is it breaks up the food into smaller pieces, and that, when they get smaller, increases their surface area to volume ratio. And with that increased surface area to volume ratio, the rest of the chemical digestion, which occurs in your stomach and a little bit in your intestine, that becomes way more efficient the smaller the particle is. So if you think back from a few weeks ago when we were doing the lab on cell size, and we had the agar cubes, the smaller the size, the higher the surface area to volume ratio, the more efficient digestion will be. So mechanical digestion and chemical digestion in your mouth. We start chewing, food goes down in your stomach, ends up in that pH2 environment down there, and then we see some of our foods breaking apart in that, a little bit more digestion occurring there, and then we decrease the size of our food particles even further, move into our small intestine, add more enzymes, more mechanical or chemical digestion, move into our large intestine, and that's where most of the absorption of food is going to take place, inside the large intestine. Anything that we can't digest and anything that we can't absorb across our intestine ends up passing out the other end as our feces. So the non-digestible material of our food, that becomes our poop. Right. So obtaining energy from the food that we eat. Enzymes. So first of all, the body must break down the large food molecules into smaller parts in order for us to be able to digest it or to absorb it into our system. And there's a number of enzymes which help us do that. We're going to look at 3 different enzymes today and the role that these enzymes play in breaking down carbohydrates, in breaking down lipids, and in breaking down proteins. So in terms of enzymes, your lecture instructor hopefully talked about this. Hopefully you've got a pretty good idea of enzymes. But if not, the simplest way of thinking about this is that enzymes can speed up chemical reactions. So they make chemical reactions occur faster by lowering what's called the activation energy of that enzyme. And the activation energy is the refers to the inherent stability in a molecule. So if you think about that bread that you had in that bologna and cheese sandwich that we made earlier, that slice of bread is fairly stable. If you take it out of its bag and put it down on your plate and watched it for 10 minutes, it's not going to start falling apart and breaking apart on you because it has some inherent stability in it. If you were to take that slice of bread and pour some water on it, you would end up with a soggy piece of bread, but it would still retain its shape. But if you put the enzyme amylase on that piece of bread and then pour some amylase on it, that bread would actually start dissolving in front of you because the enzyme breaks up the stability in the bonds and enables it to start digesting. In this case, it would be chemical digestion. So enzymes lower the activation energy and enable a reaction to occur a little bit more quickly and more efficiently. So I'm going to pause this diagram if I can. Cool. So I'm going to walk through this particular slide as it goes. We're going to start off here with this yellow capsule type thing. That represents an enzyme. So you'll notice there's a little circular depression here, a little wedge-shaped depression there, and collectively those two depressions make up the active site of the enzyme. And once I start playing this, you'll see a substrate molecule coming in. So the substrate is the molecule that is going to undergo a chemical reaction. And as the substrate comes in, it will bind to the active site, the reaction will take place. In this case, the substrate is going to break apart into two molecules, and then it's going to be released again. Enzymes don't get used up in a chemical reaction. They can be used over and over and over and over again. After the substrate gets broken down, and we see that first reaction, the next thing that we'll see is a different molecule coming in that doesn't quite match. So it's the different substrate, not the substrate for this enzyme, and that second molecule will not undergo a chemical reaction. So I'll run through this real quick, I'll start it, I'll pause it, and then I'm going to give you a different example of enzymes. All right, so we've got our enzyme. Here comes our substrate molecule coming into the reaction. It binds to the active site of the enzyme, and when it binds to the active site, our chemical reaction takes place. So now we have our two products. Our products get released, that reaction is done, and our enzyme is ready to be used again. Here we now have a second molecule coming in, but it has a different shape. It's not our target substrate, and so it can maybe bind to the enzyme, but no reaction takes place, so that one gets released, and then our enzyme is ready to go again for our target molecule. So what we just did is we showed you two different molecules coming in. One was the true substrate. That one bound to the enzyme, had a chemical reaction, and then the other molecule was the wrong molecule. It bound to the enzyme temporarily, but then it moved off again. So what would that do to a chemical reaction if you have a lot of those non-target molecules, the wrong substrate in there? So every time that non-target molecule binds to the enzyme, it's going to prevent your substrate, the true target molecule, from undergoing a chemical reaction. And I'm looking for, I found them, I'm looking for my keys. So I have two keys on my key ring. Hold them up here so you can see it. One of these keys opens my office upstairs. The other key opens pretty much every other room in this building except for the offices. And so if I were to come downstairs in the morning and try to open the door, if I put this key into the door, it'll unlock. But if I try to put my office key in, it won't unlock. If I were to pass my keys to somebody else and say, hey, can you open the door for me, assuming there was someone here, which obviously there's not, but if there was someone here, they wouldn't know which key to put in. They might put in the correct key, turn it and open the door, or they might put in the wrong key, in which case they would jiggle it, nothing would happen, they would pull it out and put in the other key. So if we think of the lock on the door as our enzyme, the key as the substrate or the target molecule, and then the reaction would be unlocking the door. If I put the correct key in, turn it, the door will unlock. If I put the incorrect key in, turn it, nothing's going to happen. I'm not going to lock the door or be able to unlock the door. But having extra keys on my key ring is going to slow down the reaction. It's going to make it take longer to open the door. So that's what the non-substrate molecules do. They are the non-target molecules. If there's a lot of those in the solution, it's going to slow down a reaction. And we can think of those non-target molecules as inhibitors. So an inhibitor is a molecule that can bind to the enzyme, and when it binds to the enzyme, it will slow down the chemical reaction because it prevents your target substrate or your target molecule from coming in. We're going to play with two different inhibitor molecules today as we run through the lab. All right, this next slide shows a little bit more on inhibiting. Let me go ahead and pause it one more time. Let me go ahead and pause this one more time. So in terms of inhibiting enzymes, well, things which can inhibit enzyme are, things that can inhibit enzymes are anything that can either bind to the active site, preventing your target molecule or your substrate from coming in, or anything that actually changes the structure of your enzyme as well. So a few weeks ago, we subjected the beets to pH outside of the optimal pH range, and that caused the beet cell membrane to break apart. As we exposed it to pH 3 environment or pH 11 environment. So same thing happens to enzymes. If they're in a pH outside of their optimal range, then the beet or the enzymes can become denatured and break apart. Changing the temperature, if we were to heat up an enzyme, just like heating up a protein or cooking an egg, we'll end up denaturing the protein, and that would cause it to stop working, or we can inhibit the enzyme. And the inhibitors are like the wrong key that I just showed you in the previous example. And there's different types of inhibitors. Some inhibitors bind to the active site, blocking the target molecule or your substrate from coming in. Other inhibitors can bind somewhere else. And if they bind somewhere else on the enzyme, it's called an allosteric inhibitor. Allosteric means other site or other location. So an allosteric inhibitor binds to your enzyme, but not at your target location, somewhere else on it. And we're going to see that going on in this, this molecule right here. So here's our active site. Here's our target enzyme coming in. Oh, actually, I'm sorry. That was the active site. And here's an allosteric inhibitor, something that comes in, alters the shape of the enzyme, alters the shape of the active site, and so our substrate can't bind and a reaction cannot occur. Some allosteric inhibitor molecules are permanent, where once they bind to the enzyme, they don't let go, and that's going to permanently shut off that enzyme. Other times, our allosteric inhibitors are temporary, and sometimes, this is actually really cool, some of our allosteric inhibitors are actually the products of a chemical reaction. So when we have a reaction occurring, our substrate goes in, breaks apart into its two products. Sometimes those products become allosteric inhibitors and bind to a different region of the enzyme, effectively turning it off. And that gives us control over how much of a product our body can make, so that we're not constantly undergoing that reaction. But if we have the enzyme and we have very few products, then the product will come out, the allosteric inhibitor comes out, that turns on the enzyme. If the enzyme is active, it can undergo a reaction, it can make more of that product, and once our concentration of the product increases in the cell, some of that product will bind to the enzyme and turn it off again. And we use up that product, its concentration goes down, the allosteric inhibitor comes out, turns on the enzyme again. It's very similar to, say, your toilet, actually. So, you know when you flush the toilet, if you have a tank toilet anyway, in the back of your toilet or in the tank, when you flush your toilet, as the water level goes down, the little float drops down. And when that float drops down, it allows your tank to refill. And then the float will go up, up, up until it reaches the top of the tank, and then it turns off the valve and no more water flows in. So it's essentially responding to need. When you flush the toilet and your water drops down, it'll fill it up again. So an allosteric enzyme sometimes will work in the same way, where the product will turn off the enzyme, kind of like the full bowl or the full tank in your toilet, and then when you flush the toilet, your product goes down, then you turn on the enzyme again in order to make more until you've got enough. I hope that example worked out for you or made sense, but that is all I've got on enzymes. So what we're going to do now, we're going to break down the three different types of macromolecules: carbohydrates, lipids, and proteins. We'll talk about where they digest in our body, which enzymes are responsible for that digestion, and then we're going to take a break from the video. I'm going to set up our experiment for us, and we're going to run through the procedures. All right, so breaking down complex carbohydrates. Complex carbohydrates, again, with respect to our diet, we're talking primarily about starch. And we talked about this earlier. Digestion of starch begins in your mouth, both with mechanical digestion with respect to chewing, and then also chemical digestion with the help of an enzyme. The first enzyme we released is called amylase, and amylase can break down that starch into maltose. And maltose is a disaccharide, or simple carbohydrate, and that maltose then is still too large for us to absorb. It's a disaccharide, but we can't absorb that across our intestine. We have to make it even smaller. So we chew up our food, we swallow the bolus of food, it hangs out in our stomach, it passes from our stomach into our large intestine, and in our in our small intestine, And in our small intestine, we're going to secrete more amylase to break down any remaining starch. And then we also secrete something called maltase. As we're transitioning from our small to large intestine, we're going to secrete maltase. And maltase can then break up that maltose, that disaccharide, into the glucose monomers, and that's where we're going to then absorb that glucose. So carbohydrates, amylase in our saliva, amylase in our small intestine, maltase in our large intestine, and that's going to then give us the glucose that we can absorb. If we break down fats, fats are a little bit trickier to break down because we need more than just an enzyme. So really not much happens to fat in our mouth, except maybe that mechanical digestion if we're chewing it up. But fat molecules tend to be very tough, tend to stick together, and so they're a little bit more difficult to break down. So the first thing that we need to do with fats is to emulsify them. And the process of emulsification simply means to separate the individual triglycerides from one another. So we secrete something called bile. Bile is produced in our liver, it's stored in our gallbladder, and it's essentially secreted into our intestine by the duodenum. And what happens here, bile is about 98% water. It's about a little less than 1% bile salt. And then you've got something called bilirubin and biliverdin in there. And there's a couple other ingredients as well. The bilirubin is a orangish yellow color. The biliverdin is a greenish color. And so the bile itself, when it's secreted, it looks green because of the biliverdin. But when it gets secreted into your intestine and begins mixing with your foods, it turns brown in color. So if you've ever wondered why your poop is brown, it's because of the bilirubin and the biliverdin mixing up together. Because the food that you eat isn't necessarily brown, but usually what comes out is. So that's what's in our bile, and also the bile salts. We're going to just be using bile salts today because that's the main part of it, which is going to emulsify our fats. And again, emulsify means to separate into smaller components. And then after we've broken up our large fat globules into triglycerides, into the individual triglycerides, then we can secrete something called lipase. And lipase is the enzyme which separates the glycerol molecule from the fatty acid tails, and that's then going to be able to enable us to absorb the components of the fat into our intestine. So bile emulsifies the fats, and then the lipase is the enzyme which will actually break apart or separate the triglyceride into the glycerol and the 3 fatty acid tails. Finally, our third macromolecule is protein. And protein digestion also doesn't begin in your mouth. Now, proteins are the largest molecules that we're going to be digesting. They're the most complex. They take the longest time to break down as well. So you would think that maybe we should actually start protein digestion in the mouth. It would be more efficient, except there's a problem with that. Other than mechanical digestion, where we're chewing up our food, we can sure chew it up, increase the surface area to volume ratio, make the enzymes more efficient, but we can't secrete an enzyme in our mouth that's going to digest the protein. Why not? Why not? Oh, yeah. Because of that. You've got a big old tongue in your mouth, and your tongue is a muscle, and your tongue is made up primarily of protein. So if you were to secrete an enzyme to break down protein in your mouth, guess what? You'll start digesting your mouth, and that would suck. So we don't want to do that. So instead, we start secreting enzymes in your stomach. Now, there's an enzyme in your stomach called pepsin, and pepsin gets secreted in your stomach at a pH 2 environment. So unlike conditions which would normally, my battery's going low. Unlike the conditions which would normally cause our proteins to denature, pepsin actually works really well in a pH 2 environment because it gets secreted in your stomach, and that's the environment, the pH environment of your stomach. So we break it down into smaller components, then we would secrete a second enzyme in your intestine called trypsin, and then a third enzyme, pepsidase. And after all of that, finally, somewhere deep in your large intestine, we can absorb the individual amino acids. So that doesn't occur until much, much later, until we're really deep in our and test it. All right, I'm going to take a break here. What we're doing in lab today, my battery's almost dead on my GoPro, that's why I need to take a break. So in lab today, we're going to be looking at how enzymes facilitate breaking down macromolecules. So let's go ahead and pause this. I'm going to charge up my camera and get this stuff set up. Okay, so I've got everything set up for you. I didn't bother filming myself setting up each test tube. You guys know how to transfer pipette. But what I've got here is experiment A, the carbohydrate test. So I've got my 4 test tubes set up, and on page, if you pull out your lab manual, on page 132, it describes kind of summarizes the entire experimental setup for each one of the test tubes. So in test tube A, right here, I put in the 4 milliliters of amylase and then 10 drops of copper sulfite. I vortexed that, I let it sit for the appropriate amount of time, 5 minutes, and then I added the 3 milliliters of starch to that. While that was waiting, I set up the remaining 3 test tubes, 4 milliliters of amylase and 3 milliliters of starch, 4 milliliters of deionized water and 3 milliliters of starch, and then 4 milliliters of DI water and 3 milliliters of maltose. Remember, starch is the complex carbohydrate. That's the macromolecule, and maltose is the simple carbohydrate, the disaccharide. And so what we're going to be doing is, in test tube A, I've got my enzyme, amylase, I've got my starch in there, but I also put in that copper sulfate, and we're going to have to figure out what that copper sulfate does. Test tube B, I've got my enzyme, amylase, I've got my starch in there, and so we would expect the amylase to work on that starch and start digesting it for this one. Test tube C, the 3rd test tube, is just deionized water and starch. I wouldn't expect there to be a reaction because there's no enzyme. And then test tube D is just maltose and deionized water. Again, we're not really going to see a reaction there. So those are the 4 test tubes for the carbohydrate test. I'm going to set these aside and we'll throw these in an incubator in just a minute. For the experimental setup for our protein test, what I ended up doing is I took a hard-boiled egg, I cut it into small pieces, and I've got my 3 test tubes set up for that one. So I've got 1 small piece of solid egg white in there, and then some deionized water. So we'll see if the water can digest the egg. Probably not. I've got a small piece of egg white and some pepsin, pepsin at a pH 2. Remember that pepsin is secreted in your stomach, and so the pH 2 might actually work well for this one, or it might not. We'll find out. And then my small piece of egg white and pepsin, and this one's set at a pH 6 environment. So we're going to compare the enzyme pepsin at pH 2 and pH 6. We'll see how that one goes over. And then finally, my 3rd setup that we have is the lipid test. And on the lipid test, I put bile salt, lipase, and milk into these various containers. So my first test tube is still perfectly clear. It's clear because we didn't put any milk in this. And on table 9-3, it's going to ask you what your starting transparency was at time 0. So that's what we're looking at right now, starting transparency at time 0. And your options for that are cloudy, somewhat clear, or clear. And for this case, for test tube A, I'm going to call that one clear, and you should too. For test tube B, I put in my milk, I put in bile salt, and deionized water. So the bile salt, remember, is an emulsifier, but I don't have any enzyme in there. We'll see how this one changes. But by the way, that one looks pretty cloudy to me. So on table 9-3, test tube B, time 0, cloudy. Test tube C, I've got milk, I've got deionized water, and I've got my enzyme lipase in there. This one has the enzyme. Let's see what happens. Ooh, but no bile salts. We're going to figure out, we're going to watch that one as well. And what do you think? I'm dealing cloudy on that one. And then finally, test tube D for table 9-3, we've got our milk in there, which makes it cloudy. It has the bile salt in there, which is the emulsifier. It has the deionized water in there, which in this case is just a filler, really. And then it has the lipase in there. So the lipase is the enzyme, bile salt is the emulsifier, and so we've got all of these guys set up on your tables. On your tables, what I want, sorry, I keep looking down here because that's where I'm watching myself. But on your tables, you'll notice that it doesn't tell you the contents of each test tube. before you scan your lab report and turn it into your instructor, make sure you tell us what the contents are for each test tube. And so you'll need that for table 9-2, 9-1, 9-2, and 9-3. And you can find all of that information on page 132. We summarize it, test tube A, test tube B, test tube C, and D for each of the experiments. So what you're going to need to do before turning this into us, copy down that information on the table. All three tables need that information. I'm going to go ahead and put these into the incubator right now. We're going to let this incubate. Oh, I've done all the vortexing. I've got everything set up. So we're going to throw this into the incubator, and in 15 minutes, I'm going to pull it out, start the camera. We're going to take a peek at how it looks. So it's been 15 minutes. I just incubated everything, or I'm sorry, I just vortexed everything. And the 4 test tubes that I want to show you are for experiment C, the lipid test. So I've got test tubes A, B, C, D from my left to my right. I'll hold each one. Test tube A was clear when we started, and it's still clear. No change for test tube A. We didn't have any milk in there, so we wouldn't expect it to change. Test tube B started off cloudy, and it's still cloudy. Test tube C, also still cloudy, but here's the cool one. Test tube D is becoming somewhat clear. Remember, we started off cloudy because we had the milk in there, but in test tube B, we also put in the bile salt and the enzyme lipase. And so the combination of bile salt and lipase starting to digest the triglycerides in the milk and starting to make it appear clear. Again, C and D were the same color when we started, and you can see that there's a bit of a difference. So we're starting to see some digestion going on. We'll check back in another 15 minutes as we take that 30-minute mark. So in table 9-3 for test tube A, clear at 15 minutes. Test tube B, cloudy. Test tube C, cloudy. Test tube D, somewhat clear. Go ahead and record those results. All right, gang, we are at 30 minutes, and I just took everything out, vortexed it one more time. We have to check the transparency for our experiment C, the milk test. And so, again, let's take a peek. Test tube A, no change, started clear, it's still clear, so we wouldn't expect anything to happen in there because, well, we didn't have any milk, so it's not going to change color. Our test tube B started off cloudy, it's still cloudy, no change with test tube B. Test tube C, still cloudy, again, no change with that one. And then if we look at test tube D, it's a little bit clearer than it was last time. So our transparency is getting a little bit more transparent. The fats in the milk, the triglycerides, are breaking down a little bit more. And I'm going to go ahead and put these back in the water bath at 37 degrees. We'll let it run for 15 more minutes, and then we will look at our results. So let's take a look at the results. As this is cooking, incubating for the next 15 minutes, I'll talk about real quickly how we're going to interpret our results. And then after that, I'll run everything through. I'll show you the pictures or the videos at the end of everything, and then summarize our lab for us. So the first test that we're going to do is we're going to test for the presence or absence of starch. And we do that by using something called a depression plate. Now the depression plate, Ooh, all right, sorry about that. Pretty strong, but the depression plate has a lot of little wells in it, and what we're going to do is we're going to take our solution from experiment A, the first experiment, the carbohydrate test, and we're going to put a little bit of We're going to put a little bit of each solution into each one of the 4 wells. So a little bit from test tube A, a little bit from test tube B, test tube C, and test tube D. And then we're going to use iodine. And iodine actually changes color in response to or in the presence of starch. So iodine is usually a nice amber color. It looks like maple syrup. But if there's starch present, the iodine turns dark blue or black in color, almost purple or black in color. So it's a very rapid transition, and I'll go ahead and show you that in just a little bit. So if we test positive for starch, it's going to turn black. If we have negative for starch, so there's no starch in there, it's going to be amber. And if there's some starch in there, so there's a little bit of starch, it's going to be amber with some particulates or some dark specks in the bottom of that. So once we get through this, in another 15 minutes, we'll go ahead and look at that, and I'll show you what the results are. In the meantime, think about, well, what test tube do we know should test positive for starch? Yeah, the test tube that we only put starch and water in. Which one do we know will test negative for starch? Yeah, the test tube that we put the maltose in, but no starch. So those will be, we know one will be positive, we know the other is going to be negative, and then we have to think about what happened with test tube A, where we had the, where we did put in the starch, but we also put in the enzyme amylase, but we had copper sulfate. Or test tube B, where we had the starch and the amylase, but no copper sulfate. So those are really the two interesting test tubes, test tube A and test tube B. Test tube C and D, those are our controls. So that's what we know what a positive is going to look like and what a negative will look like. Once we're done with that, we're going to save those 4 test tubes and we're going to add something called Benedict's solution to them. And Benedict's solution is that solution where we finally get to wear our safety goggles, or I do anyway, and that's because Benedict's solution, if it gets in your eyes, yeah, not a good thing on that one. So with Benedict's solution, all 4 of our test tubes are going to start off with the blue color because Benedict's solution is blue. But the cool thing is when you put Benedict's solution into boiling water, and I've got a beaker of water that's heating up, about to boil. When you put it into boiling water, it changes color. Benedict's tests for simple sugars, monosaccharides or disaccharides. And we know that in one of our 4 test tubes, where we only put maltose in, test tube C, the 4th one, test tube D, we know that that one's going to test positive. And a positive result turns bright orange, like carrot juice. We know that at least one will be negative for starch, or for maltose. That's test tube C because we didn't put any in and we didn't put any enzyme in there. So that one is going to be blue in color. Test tube A and B, well, we're going to see whether or not the amylase was able to break down the starch and convert it into maltose. And if it does, it'll be orange. If there's a little bit of maltose in there, a little bit of a breakdown, it'll be kind of an intermediate green color. So we're going to look for those color changes once we boil those up. And then the third test, the protein test, this one's a little bit more difficult to interpret. Remember, we started by putting a piece of egg white into the test tube. We put in either deionized water, we put in pepsin at a pH 2 or pepsin at a pH 6. And what we're going to look for there is some cloudiness to the water. The water might get a little bit murky, or not the water, but the solution might get a little bit murky. It might get a little bit cloudy. We're not going to see an appreciable decrease in the size of the egg. I would have had to weigh the egg with a scale and measure exactly how much each weighs in order to determine whether the egg is getting smaller. So what we're going to look for is whether the solution is getting somewhat cloudy. And if the solution is getting somewhat cloudy, what that means is that the protein is breaking down, chemically being digested, and that the amino acids are breaking off of that and coming out, the amino acids are breaking down, coming into the solution and causing the solution to become a little bit murky, a little bit cloudy, a little bit whitish in color. So those are the 3 results that we're going to be testing for. I've got 4 different tests: iodine, test for starch; Benedict's, test for simple sugar; and then we're going to do the color change, clear versus cloudy, test for starch; Benedict's, test for simple sugar; and then we're going to do the color change, clear versus cloudy on the milk, and then also the lipid test. Now, the milk is pretty cool. So we added the bile salt, and remember that the function of the bile salt is to emulsify the fats. So we were emulsifying the triglycerides, making them smaller, and then the lipase is the enzyme which breaks down the lipids. So those 2 together is what we put in the 4th test tube, A, B, C, D, and that's where we started seeing the transition from cloudy to clear. If we only have the bile salts in there, well, we didn't see any change. If we only had the lipase in there without the bile salt, we didn't see any change there either, at least not yet. But I still have about 7 more minutes to go before we can pull these out and vortex them one last time and then run these tests. So I'm going to stop the camera. That's what we're up against right now. I'm going to leave this one up as I'm walking away, and then we'll talk about our study guide for your quiz next week as we come back in. All right, gang, so we are at our last 30 minutes, 45 minutes, and I've got this set up. I'm going to go ahead and put my Benedicts into the water bath, but I want to show you to start off with where we're at. They are all blue in color because I added the Benedict solution to that. I've already pulled off the solutions and put them into the depression plate for the iodine test, labeled A, B, C, and D. And let me go ahead and just set this up over here. These 4 go into the boiling water. We're going to let those sit for a few minutes until they go. And now let's take a look at the iodine test. Take the iodine. And test tube A, see how it tested positive for starch. Test tube B, test tube C, and test tube D. So here are our results for the iodine test. Remember, C and D were our controls. In test tube C right here, we put in starch, but no amylase, no enzyme to break it down. So we would expect a positive result from that one, and that's what we got. For test tube B, we didn't put any starch into it. We only put in the maltose, or this simple carbohydrate. And so that one tests negative for starch. It's an amber color. Test tube A, it's not quite as dark. I'm not sure how it's showing up on the video, but it's not quite as dark as test tube C. So there was a little bit of breakdown, but not much. And then test tube B looks almost the same as D. So the amylase that we put into test tube B completely broke down the starch in that one and converted it to a simple sugar, or a disaccharide, maltose. All right, I'm going to go ahead and set this up. All right, for our other results, for the protein test, we had our 3 separate test tubes that we had the pepsin in. We had, first of all, test tube A, which had the piece of egg white and the deionized water. And I'm not sure if it's showing up, but that solution is perfectly clear. The egg did not break down at all. Test tube B, which has the pepsin at a pH 2 and our piece of egg in there. I'm not sure if you can see that, but that solution's turned pretty cloudy. It's pretty milky in there, and so that's an indication that the egg has been broken down, or at least some of the egg has been broken down, and the amino acids are now suspended in that solution. So this is test tube B with the pepsin at a pH 2. Our third test tube was test tube C, pepsin at a pH of 6, and this one is pretty clear still. So the pH 6 pepsin really didn't do anything to break down the egg. The pepsin at a pH 2 had a significant amount of breakdown. That's why the solution was cloudy. Pepsin at a pH 6, not really any change on that one at all. All right, and finally, let's take a look at our lipid test for our milk. Test tube A. Again, this one's been clear all along because we only added the water to this one. We didn't put any milk in it, so it stayed perfectly clear. Test tube B, which had the milk, but it didn't have both the bile salt and the enzyme. It just had one. It started off cloudy. It's still cloudy, so you're going to want to record that for time 45. Test tube C, also still cloudy. And then test tube B, or D, which had the milk and the bile salt and the lipase. Check that out. It's as clear as test tube A. So the milk is completely broken down. It's turned completely transparent, and that's because we've completely broken down the triglycerides and the fats in there. So we've completely digested the fat, broke the triglycerides down into the glycerol and the fatty acids, and so that solution has become clear. And we needed both the bile salt and the enzyme lipase in order to do that. So these solutions were all blue when I put them in a few minutes ago, and I can go ahead and take them out already. So the color change that we see here, and these are not in order, by the way, but the color change that we see here is now an indication that the Benedict solution has done its job, or the Benedict test has done its job. And if we wanted to go ahead and put these in order based on the strongest or the most maltose in there, our simple carbohydrate, we can start off with this one. This is a really strong positive test result, and that one is going to be test tube D, the test tube where we added only the starch or the maltose to it. So that's where we added the maltose, the simple sugar, to it. Test tube C, nearly blue, so that's an indication that there was no simple carbohydrates in there at all, and that's the one that tested positive for starch, but it's testing negative for our simple carbohydrates. Now, test tubes A and B are a little bit different, and I'm going to go ahead and put them this way. The reason I'm going to put them in this order is, remember that when we were doing our iodine test, we added the starch, we added the copper sulfate, and then we added the, we added the amylase, and then the copper sulfate, and then we added the starch to it afterwards. But we didn't see very much breakdown of the starch based on our iodine test. Remember, that was the test tube A in the far left, and it was still very dark. And so it's an indication that there's a little bit of simple sugar in there, but not a whole lot. Test tube B, which is showing an orange color, this one actually has a significant amount of maltose in it, or the disaccharide. And that actually makes sense if we compare it to the maltose, or the IKI, the iodine test as well, because remember, test tube B, even though we put starch in, we also had amylase, and the amylase broke down the starch and converted it into maltose, which is what we're seeing here. So I'm going to put these two together with the iodine test, and I'm going to go ahead and narrate that one more time for us. So let's start with test tube A. Test tube A, positive for starch, and just a slight positive, little bit of green there with respect to having the maltose in it, the disaccharide. Test tube B, we added starch at the beginning, but we also added the amylase, and the amylase broke down the starch, so mostly negative for starch, and positive for maltose, right there. Test tube C, we only added starch, so we didn't have any amylase, any enzyme in there, positive for starch, negative for maltose. And test tube D, which we added the maltose, well, negative for starch, but our positive for maltose. So these are the colors that we're going to want to put into our table in terms of the Benedict's color. So if we're looking at the IKI test results, it's going to be black or blue, kind of a light brown, and then black and blue, and then light brown or yellow right here, and then positive or negative for starch, positive, slight positive, positive, negative. The Benedict's color, it's going to be kind of a green, orange, blue, orange. And so that's going to give us that. And again, that will also tell us positive for negative for maltose on there.
All right, everybody, I'm back at home, and I am so sorry for the quality or lack thereof of those videos, I should say. I had to break it up into 4 separate sections, completely not my intent. But what I'm going to do now is I'm just going to go over the results one more time, hopefully really quickly. I'm going to pull up the lab report and fill in a few of the blanks and talk about what to do for your submission for us next week. So let me go ahead and open up the lab manual. I've got a copy of it on my computer, and we're just going to talk about the results real quick. So let's go ahead and do that. And so this is what was our experimental setup over here. So we had the carbohydrate test where we put in the amylase, the copper sulfate. In case you are wondering, why didn't that test tube change very much? Well, it turns out that the copper sulfate is an inhibitor. And so what the copper sulfate did is it inhibited the amylase from doing its job on the starch. So we didn't see very much digestion in the starch, That's why we had a positive test result for our starch and a negative or mostly negative test result for maltose when we were doing the carbohydrate test and the Benedict's test. Test tube B, we had our amylase and starch. That one did exactly what we would expect it to do. The amylase broke down the starch, so we were mostly negative for starch during the iodine test, IKI, and we were mostly positive for maltose or simple carbohydrate during the Benedict's test. Test tube C, we only had starch, so strong positive with IKI, negative or blue with the Benedict's. And then finally, test tube D, we had our deionized water and maltose, which tested negative for starch. We didn't put any in, but positive for the simple carbohydrate or maltose. So that one turned the bright orange, kind of the carrot color, carrot juice color. Our protein test, we put egg white into each. Test tube A, we had the deionized water. Obviously no change because water is not going to break down the protein. Test tube B, with our pepsin at pH 2, that's where we ended up seeing the milky solution. So that's the solution that turned fairly milky for us, or we saw the amino acids breaking down. And then finally, test tube C with pepsin at a pH 6, not much change there either. And we didn't get much change simply because the pepsin doesn't work well at a pH 6 environment. Remember, your stomach as a pH 2 environment, pepsin is secreted in your stomach, so it actually works well at a pH 2 environment. And finally, the lipid test. We had test tube A, bile salt, water, and lipase, but no milk. That one started off clear and remained clear. Test tube B, milk, bile salt, and deionized water. Even with the bile salt, which would emulsify the lipid, or the triglyceride, I'm sorry, in that case, it did not break down. So it remained kind of cloudy or milky in color. Test tube C, we had the milk, we had the deionized water, we had the lipase, but no bile salt. Again, the triglycerides were too large to be broken down without emulsifying them. Without the bile salt, we didn't emulsify it, so that one didn't break down. And test tube D was our big winner there. That's where we had the milk, the bile salt, which would emulsify the triglyceride, the deionized water. Again, in all of these, the DI water is essentially just a filler to increase the volume. And then we had our lipase. So the bile salts emulsified the triglyceride, and then the lipase separated the glycerol from the fatty acid, and that's why that test tube was able to turn clear after the 45 minutes in the water bath. So let's take a look at our results here. So again, remember, you're going to want to fill out. Okay, we're going to continue with this. So remember, what we're going to need to do is fill out the contents of each one of these test tubes before turning it into us. So go back to the table on page 132 and tell us what is in each one of these test tubes. So for the IKI color test at the end, test tube number A, it turned out that that was kind of a dark blue or black. I'm just going to put black because that one still had a lot of starch in it, a little bit of breakdown, but the copper sulfate prevented that from breaking down fully. And so we had starch present and a positive there. For test tube B, we were light brown. And then no starch. Test tube C, that was our control, black. Positive. And test tube D, that was again light brown. And a negative there. In terms of the Benedict's color test that we had, this one was kind of orange. I'm sorry. I'm sorry, I messed up there. This one was green. And so the green or the yellow was a plus minus. And then we had orange, plus. We had blue, negative, and red. A plus for that one. So those are our results those are our results for the carbohydrate test. If we were to scroll down to the next one, and it's not scrolling for me. All right. So again, for table 9-2, we want to fill out the contents of each test tube. You're going to want to do that. And then in terms of the clarity of our solution, we were clear, cloudy, and then clear. So in terms of no change in the solution clarity, we had only protein but no amino acid. For test tube B, we had protein and also amino acid, so plus minus. And then for content C, it was just a positive. Those are the results there. I'm going to scroll down a little bit here if I can. Over here, our transparency was clear. Cloudy. Cloudy and cloudy. Clear. And then contents B, this one remained cloudy. And then really, the one that was most interesting for us was test tube, the last one, which was clear and then somewhat cloudy. Let me pause this one more time. All right, I finished up the table here. And so again, clear, clear, clear all the way across. Don't forget, you're going to want to put the contents of each one of the test tubes in there before you submit it for us. And where I left off with was test tube D, which was cloudy initially. It became somewhat clear at 15 minutes, somewhat clear at 30 minutes, and then by 45 minutes it was clear. So cloudy represents that triglycerides are still present. Clear represents that they're digested. Well, we didn't put any into test tube A, so that one's a negative. Test tube B is going to be a positive. Test tube C is a positive. And then by the end of the 45 minutes, test tube D, which had the milk, it had the bile salt, it had the enzyme lipase in it, and then the DI water is the fill. Again, that one became clear, so that one will be a negative for us. So those are the results for tables 9-1, 9-2, 9-3. Let's go ahead and clear all of this. And then what I want to do is go through the last few questions for us. So in terms of the lab report questions, number one, I'm actually going to answer these for you because it's really question number 5 that we're interested in for this lab in terms of you filling it all out. So number 1, where does digestion begin in your body? Well, if you watch the videos, digestion begins in your mouth. And so you can simply answer that with your mouth. And then we have both mechanical digestion, which is chewing, and the chemical digestion with the enzyme amylase. Question number 2, what is the first macromolecule that your body breaks down? Well, that's going to be starch. And then the enzyme for that one is, again, amylase. And that's located in your saliva. So based on the protein test, what is the pH inside of your stomach? So with that, well, we had 2 different pepsin pHs. We did pH 2, which actually worked pretty well, and we had pH 6, which didn't work very well. So remember that pepsin is secreted in your stomach, and if the pepsin at the pH 2 digested the protein, well, that would be a good place to start in terms of answering that question. Do you think proteins are completely broken down in your stomach? I'll let you think about that one. But did we break down the egg completely or not? Remember, if you go back over the PowerPoint and the slides, there's additional enzymes which get secreted in your intestine as well. We've got pepsin, trypsin, and pepsidase are 3 different enzymes, and they get secreted in your stomach and then also along in your intestine. So I'll let you answer that one on your own, on that one. And then explain the role of bile salts. So with respect to bile salts, remember that bile salts emulsify lipids. So that is their role, to emulsify lipids. And here's the big question. This is the one really where I'm going to spend most of my time grading this lab report. I'll read the statement to you, and then I'll tell you what I want you to do with this one. So in terms of this question, what the question says is, orlistat is a prescription drug that acts as an inhibitor for lipase, which is the enzyme used to digest fat. That's a true statement. You could get a prescription for orlistat, and it will inhibit lipase in terms of its ability to do its job, which is to digest lipids. And we ask you to briefly design an experiment to test this statement using 2 test tubes. Realize you want to use only 2 test tubes. Don't give me 3, don't give me 4, only 2 test tubes. You should include a list of all materials. So what I would want you to do is start with the word materials. And then give me a list of what those materials are. I encourage you to use one of the 3 experiments that we did in lab or that we presented to you, but choose the one that actually deals with lipids and then copy down that list of materials. Realize also you're going to have to add one more thing to that, and that would be the prescription drug, orlistat. And then write out your step-by-step procedures. Do this as if you were writing another lab report. So after you list your materials, the procedures, step 1, step 2, step 3, tell us the exact quantities of each substance that you'd want us to add to test tube number 1 and the exact quantities that you would want us to add to test tube number 2. And then what do we do with those? Do we vortex them? Do we not? Do we put them in a water bath? Did we not? Did you include the water bath and the vortex in your materials? You should have. So after you do that, give us the procedures and then give us an explanation of your predicted results. Obviously, we're not going to conduct this experiment, but if you did, what would you expect your results to be with respect to clear or cloudy in your solution? So it might be helpful to write out a table in your results. Your table can match one of the tables in your lab report where you have your experiment running for a certain period of time. You take it out, you vortex it, you look at it every 15 minutes. What are your results going to be? And then finally, conclude your experimental write-up by answering the following question. If orlistat is an inhibitor for lipase, what conclusions could you make about digesting and absorbing fatty foods when taking this drug? So again, in summary, give us a list of your materials, give us a list of your step-by-step procedures, tell us what your expected results would be. You can even write the words expected results, colon, and then conclude by answering that statement. If orlistat is an inhibitor to lipase, what conclusions could you make about digesting and absorbing fatty foods? All right, that's all I've got for you with this one. Hopefully you're able to do this lab, and if not, come up to the ConferZoom sessions. I'll be holding one. Every other lab instructor is going to be holding a ConferZoom as well this week for your lab section. Sign into that. Ask us any questions that you need. Otherwise, stay safe. Good luck with social distancing. That's it.